Well, I'd like to welcome you all to the second in the series from the Center for Computational Biology's seminars by industry leaders in genomics and related areas. I'm really pleased to see such a great turnout here today, and no doubt it's in large part because of the speaker that we have today. Anne Wojcicki, one of the founders of 23andMe, did her training in biology at Yale, and has since that time served many different roles, including one of the founders of 23andMe, one of the very first companies in the sector for personal genomics. Now, those of you that have TA'd anything related to biology or genetics can appreciate the difficulty of communicating to a naive audience the implication of a single mutation. The goal of 23andMe is to interpret the variation of the human genome and explain it to consumers widely. So this is a huge scientific challenge, educational challenge, and increasingly regulatory challenge.
Yeah, yes.
So I'm sure we'll hear a great deal about that today. So Anne, welcome. It's great to have you here at Berkeley.
Thank you. Thank you. It's great to be here. I was actually telling them earlier that I grew up on Stanford campus, and I've always lived right next to Stanford. But my parents are actually, to turn this on. But my parents are actually from Berkeley, they went to Berkeley. My father has his graduate degree from here. My mother has her undergrad and graduate degree from here. So I felt like I was always sort of underrepresented in my Berkeley side, since I'm a true heterozygote here. But and actually the company is unfortunately a little over Stanford represented. So we can consider this also a job luring talk to help balance it out and represent the true heterozygous nature of the company. So we're going to talk. We're going to go through essentially. I left the presentation relatively simple because the most interesting part of the company really is the demo, and I think then we'll leave it relatively interactive. So here's the agenda, which we'll ignore. So one of the #1 questions that we get is, why did we start this company? And just to get a show of hands, how many people have ever been in a for-profit world? Oh, that's good. So some of you might be able to relate to this, but when people ask us why we started the company, we usually say it was because of this. We were so frustrated with what was happening. And my background, again, like Jasper said, I did a graduate degree did undergrad at Yale, and then went straight into the investing area. And in part because I thought investing in biotech and pharma companies was going to be so fascinating, and it was really going to be a way of changing the world. And all I discovered was that it was just horribly, horribly inefficient. And it was tragic to me that there was all these people who really want to do good things in healthcare, and they're just not. It's just, the system is so frustrating that they weren't able to do it. And my co-founder, Linda, she came really from being within the industry and working with a lot of academics. And I'm sure, as a lot of you know, there's a lot of really frustrating aspects about the grant writing process and publications, and there's all these things that just make it really inefficient. And at the end of the day, I feel like I'm 34, almost 35 now. I spend a lot of time in hospitals just because I like hospitals and sick people. I don't want to have that same fate. So I want to have better healthcare, and especially on areas like pharmacogenomics, like I just can't take it. I absolutely can't take it. And so so here's some statistics for people who don't know anything about it. Do you guys have any idea how much money is spent collectively by the major pharma companies on research? Any guesses out there? How much?
30 billion.
30 billion?
A year.
How much?
GSK, I think it's like 4 billion a year.
Yeah. Yeah. So no, it's a lot. I don't know. I mean, you can think about all your grants now and think about maybe you should be asking for more money. It's about 58 billion a year got spent last year in the total pharma industry. So it's just an amazing amount of money. And then roughly the cost to develop a drug, and again, you start looking at these things of how much it's actually increased over time. So, and these are all statistics from the pharma industry, when they start to justify why it costs so much money. Like they're almost proud of this fact, like, oh, it costs so much money to develop a drug. And we always found that a little crazy. And so much time. Any idea how long it takes to develop? Yeah. Oh, you guys are good. 10 to 15 years. So, and you think about, all this money, where's it going? This is where it gets to be really kind of sad, where if we spent $58 billion last year and we got 19 new chemical entities, that's that's not a great return on investment. So none of you should ever invest in biotech. So it's just remarkably inefficient. And people used to ask on the investing side all the time that, if it was really just a question of brains, Pfizer would never fail, because Pfizer has all the money. Pfizer could do anything that they want. So it's not just a question of brains. It really became a question of data. And when there was a couple of people, actually I had a really great dinner with a guy named Marcus Stoffel, who some of you might know, who he did this kind of insane project, which I always love insane projects. And he went to this island in Micronesia, and he actually genotyped. He got access to the entire island and compliance from the entire island, and he genotyped everyone. And I think that they had something like, there was almost 80% obesity, and of that population, about 60% had type 2 diabetes. And he's like, I have so much data, so much data I don't really know what to do, but not really enough data to make sense of it. And it became one of those things like, all this money gets spent. Like, if you look around the room, and if you look around the whole world, like we all have the answers to all these questions. We just need to bring all the people together, and everybody, you have your genome, you have your phenotype. If we actually all put that information together, we could solve a lot of this information. And a lot of the reasons why it wasn't happening is, you have all the patent issues, and you have, again, publishing issues and grant issues. There's all kinds of reasons why people are not sharing information. And at the same time, like, that's not necessarily in the interest of any of us. If any of you have ever been sick, or if any of you have relatives who've been sick or had an adverse event, that, like, I don't care. Like, I just want all this information to come together. So, Linda and I, when we met, we realized a couple things had happened. One, the price of this genotyping technology had really dropped. So Linda and I had both been involved with Affymetrix for a really long time. It had gotten really cheap, and it was also great because then there was finally competition. So Illumina had popped in. You started getting all the next generation sequencing technologies, which are really exciting. And as well as the fact that you have things like YouTube, MySpace. People are really used to putting information online, and YouTube took down an entire industry, really fast. And I don't think a lot of people realize. I mean, we had a lunch the other day with a bunch of major, heads of movie studios, and they were saying, it's a real threat to them, because if you can't get their latest movie, I don't know, you just watch, a Turkish dance video on YouTube, and it kind of equalized it, and so kind of. But that was their point. Like, that's a threat to them. Like, that just, like, YouTube eliminated distribution. Like, it equalized the playing field. And what we were really hoping to do with 23andMe is to create a revolution where individuals can put their information, you can get access to your genetic information, you can put that information online, you can put your phenotype information online, again, in an anonymous, private way, but be part of a community that is really going to then connect to the entire research world and is going to really start to drive discoveries. So 23andMe, some of the principles that we have about 23andMe, fundamentally, we believe very, very strongly you have the right to get access to your genetic information. And the really rewarding thing that has been, even when I was on the investing side, when you would talk to some of the companies who are actually even patent holders in these areas, everyone believes that you have the right to get access to your own genetic information. And it's just a fundamental right. So it's going to be interesting to see how it plays out with all these people patenting SNPs and patenting genes. But fundamentally, people have not disagreed with us that it is your right to get access to your genetic information. Oops. And second, we believe really strongly that it's also your right to share this information. That if it's your data and you want to compare it to other people, you should have that ability to. And one of the things that we've been really surprised about with 23andMe is that people really want to share. Like, it's actually shocking. And it kind of overthrew some of our systems that we built because we didn't actually plan on people sharing that much. And we have one person actually recently who was really looking for someone else in their haplogroup. And so she just typed in all the names, to see who she could share to. And she sent out, a few hundred invitations. And almost, I don't know, something like 70% of people accepted. So people are pretty open to the idea of just sharing with this random stranger. And people seem to really enjoy it. And one of the questions that we get all the time is that people want to know, is there someone out there in that database that I'm distantly related to? And that's a pretty cool question. So it's one of the things that we'll start to do as the database gets bigger and bigger. But people clearly are interested in connecting through their genetic information. So we recently launched something called 23andMe. 23andMe is really based about you getting access to your own information. 23andMe is really where we're hoping individuals come together and they fill out surveys and they input phenotype information about themselves. So one of the premises that we have here is that individuals have that ability and have that right also to input phenotype information about themselves. And we get questions actually from a lot of physicians where some people will input, Google Health just came out with their medical health profile, and there's some physicians who will say, like, does an individual actually have that right to put their medical information online if they wanted to? And it's fascinating to me that that's been sort of a question, like, do you have the ability to is that your right to social network based on a specific disease or a phenotype? And we strongly believe that if you want to put information about you, you have that ability to. There's a great website called Patients Like Me, and it started with three brothers who had one who had ALS, and that patients really put in very specific information about themselves, and again they were getting that response from people saying, but how do they know this information? How do they accurately put this in? And again, we have probably, maybe it's naive, but we have a stronger faith that the average individual can accurately input information about themselves. We also really want to democratize research. And one of the things that has, since we launched 23andMe, we've had really almost an outcry of individuals from the rare disease community, where individuals really want to have a platform to come together and form a community. And we had one person who said his daughter was born and she has a muscular disorder, and she's the 13th person in California to have it. And there's no physician in the Bay Area that can be an expert if there's only 13 people in California. So she's really looking to connect with other families that are out there, but there's no platform for it. And so one of the things that 23andMe is really hoping to do is for a lot of these conditions where there's underfunding or there's a lack of researchers doing interest, give them a platform to communicate. And then at some point, we hope to then partner with researchers where we can say, hey, we're serving up to you, here's 500 individuals with this condition, here's their phenotype information, here's their genotype, let's see if we can actually find anything about them. So our end goal is really to create the world's largest database of genotype, phenotype information. And it's not just for our purposes. I think 23andMe really sees ourselves as the liaison between consumers and the entire research world, where how can we actually facilitate that connection, where if researchers are trying to do a lot of good in the world and consumers really want to see change happen, there's almost a disconnect. How can we facilitate that and make that a lot more efficient, a lot easier? So, some of our goals with this is if we are collecting all this genotype, phenotype information, what do you do with this? And our end goal too is really to start to translate this information into some kind of clinical practice, or figure out how does it actually end up improving your lives. And it could be as benign as, one of the things that we ask people is, when you eat asparagus, do you smell it in your urine? And I don't know if there's anyone who has no idea what I'm talking about. Anyone?
Yes.
Okay, yeah. So, well, the great thing is when people don't know what you're talking about and they just what are you talking, do I have to sniff it? Do I have to really get down in it? And so, but there's some people who have real interest, kind of niche interest. Almost every academic out there has some kind of niche interest that they're interested in, or almost all of you clearly have something unusual about yourselves. Like everyone who comes in our office has oh yeah, Michael Eisen was showing us one of his weird tricks the other day. I think he's double jointed, or he has something interesting about him. I don't remember what. But everyone has something. But so one of our goals is to start to translate. What actually does that mean? If we have these 580,000 SNPs, well, what does this actually mean? And can we actually start to correlate this information with a better outcome? So instead of, if you have a new child and the child's sick and the doctor gives you penicillin, can you know ahead of time if your child is likely to be allergic to penicillin or not? Or peanut allergy, which is one of the big epidemics now, is there a way that this information could actually help you know ahead of time that your child is allergic to peanuts? So again, our goal is really to focus, and this is what we can't do alone, where we have to work with the academic world. How do we actually go through this translational process? And this is an area where it's really undefined. What is the definition? We can clearly define analytical validity, but how do we define clinical validity? And there's a bunch of type 2 diabetes markers out there now. I don't know, at what point do we define that those are clinically valid? So that's one of the things that 23andMe is really focused on. And we're really focused on working with all the community. So Linda and I spent a lot of time with researchers. We have a really spectacular group of scientists. 2 guys essentially were the first 2 employees, essentially co-founded the company with us, who came out of the Stanford BMI program. We recently hired Joanna Mountain, who was, not recently, almost a year ago now, who is a professor at Stanford. We just got Nick Erickson, who's a mathematician. So we've been building out a team, and I think we're really focused on achieving this. So I think at this point, and the end goal, obviously, is to dramatically accelerate the pace of research. I'm going to go back to the interesting problems afterwards. And I'm going to go to, so I think at this point, we'll just go to the account, because I bet a lot of you have not seen. Have any of you been genotyped, actually? Ooh, 1. And what do you think?
I've got the kit, but I haven't sent it back to you guys yet.
Oh, oh my God. You're not alone. We've actually been surprised. So here's the basic website. Let me log in. So we're just going to go through my data. So this is the sort of the opening page, and we made a very intentional decision when we started this company that we were not a healthcare company, and we're not an ancestry company, and we're not a social networking company. That we're about your genetics, and you can do anything you want with your genetic information. And you want to make art, go for it. If you want to make ringtones out of it, which one person in our company really wants to do, you can do that. So we don't want to limit you. Your genetic information is really cool. And we kind of want to encourage that sort of nerdy, like, yeah, your genes are cool, man. And so it's one of the things that's a challenge for us, because we meet people all the time. I was just telling the group earlier, we meet people all the time who are like, oh, do you want to, you're going to genetically modify babies, aren't you? And we're actually really not. So resoundingly. So we separate the website right now into, it's somewhat divided a little bit into My Gene Journal, which is where you're able to link your genetic profile to all the latest research that's come out there. And then we have a family and friends section, which is where you can start to compare yourself. And again, it's been a really fun thing. If you have siblings or you have cousins and you want to see in what area are you the same, it's pretty interesting. And then we have the Ancestry side, which is really led up by Joanna Mountain and her team. And again, exploring what are your roots. And this is an area that we can really start to delve a lot more into. We just launched something called Chromosome Painting, where any of you who have mixed backgrounds, and you can start to break down and see, what parts of, if you're looking at your chromosome, what parts are from Asia, what parts are from Africa, what parts are from Europe, and we'll show you one demo of that. So I'll start with Gene Journal, because it's the part that gets the most attention, because it's a little bit of the most controversial. So we had a very interesting debate when we started the company, where we wanted to be responsible. And so we were very cautious about what kind of information we were going to present. So we originally only launched with 17 Gene Journal articles. And you can see now we have about 75. And what happened is we had, Mike Eisen's on our SAB. We had all these individuals, all these esteemed professors, like, oh, we have to be cautious. We can't put out too much information. And then we launched. We gave them their 17. And the next scientific advisory board meeting, they were like drug addicts, like, give me more, like, I just want more. And we actually didn't do anything in the meeting except for go through various papers, and then everybody would look at their own account and be like, do I have that? Like, so it was a total waste of time, but it also, it was so entertaining, because they were like, like, throw out all the rules, like, just give people the information. Like, this information is so fascinating. It's your tax dollars at work. You want to start to know, what does this information mean? So we tried to come up with, what we kind of see as, a dumbed down version of the movie rating system. How can we actually show people that, because we realized the average person doesn't necessarily know that there's, some research that was really well done and some research that's probably a little bit earlier stage. And so we try to, and this is a revolutionary concept for people where they're like, oh, something in science isn't necessarily fact. Like, wow, I never realized that. So we set up the star rating. For you guys, it's so obvious, but the rest of the world doesn't know. So the star rating system is the research confidence. And 4 stars means that it's in the established research section. And that's really where, we can just show all the established research first. That's where we feel pretty uniformly there is academic consensus that these associations are true. And it's very different than saying that this is clinically meaningful, but we believe that these associations are true. I don't necessarily know that if you look at your, type 2 diabetes and you're high on that, does it actually have a clinical meaning for you? And that's really the next step of the company. So these are the ones that we have now. Macular degeneration is really, I'll do type 2 diabetes is sort of our most complicated because it is so many, let's see. It's actually, I think it's 8 or 9. We'll see. So the way we set this up is that we try to give people a little bit basic information about, what is type 2 diabetes. And then we scroll down, and then you can start to see, OK, here is my data. So, or here's sort of the summary of it, what we call the odds calculator. So this is me compared to the average. So I'm 4.3 out of 100. Average is 4.9. It is adjusted for, based on whatever this research shows. I can pick my ancestry. So I'm European descent. One of the big problems that we have is that, as I don't know how many of you know, is most research is done in Caucasian. So it's one of the things that we really hope to rectify over time is that as we start to get information from mixed populations, it's really hard to just classify someone as European, Asian, or African because so many people are mixed now. So what does that actually mean? So we're really hoping to start to expand that out. And then I can choose my age range. And then this is actually the genetic information down here. So we also try to emphasize to people here, that your genes are not everything. And this is what we see as the really positive message from Gattaca. Gattaca show your genes aren't everything. And most people don't get that point. But we try. It's actually a very good point. And one of the things we were really happy, Francis Collins was at the World Economic Forum talking about, how some of this information is a lot like cholesterol. That if I tell you have high cholesterol, it doesn't mean you're going to die of a heart attack. It just means you have high cholesterol. And I can't even tell you, without knowing your family history, if high cholesterol means anything. Is it like you might just be from a family that has high cholesterol but lives till they're 105. Or you might have high cholesterol and be from a family that dies at 45. You just don't know. So it's just information to include in your whole medical profile. So what we show here is then, this is my genetic profile. You can see here's TCF7L2, which is one of the big type 2 diabetes markers that have been found. I can click on this. We tried to make this very consumer friendly, but also nerdy for all of us who want this information. So here's the information. I can scroll down. I can see the RS number. I can see all the citations. If I want more, I can click on any of these and keep reading more. So we can then, then we go into other sections in terms of like, how does it work? With the basics of type 2 diabetes, what was the timeline, when was type 2 diabetes actually discovered, some of the historical information. And we also have a section called MD's perspective, which we don't do for all the gene journals, but we realized that some people really want to know, I found I was at higher risk for type 2 diabetes, what does that mean? And so we clicked here, and we have some type 2 diabetes. This is actually with Fro's Papa, who's MD, PhD at UCSF. And you can start to read, what does this actually mean? And then resources and the tech report. The tech report is probably, again, it's really, I don't know how many of you would ever really want to read it, but you might if you were into type 2 diabetes. So that's there. And then we go to some of the fun ones. So we have things like bitter taste perception. So am I likely to, I don't know how many of you have ever done the PTC paper, but I always find it fascinating because some people just think it's a piece of paper and they stick it on their tongue and they're like, oh, what was that? And they're scratching their tongue. But some people clearly like Brussels sprouts and some people don't. And again, you're never going to get an NIH grant to study this, but it's something that everybody can relate to. Some people, what we actually found is that there is a small percent of the population that abhors cucumbers, like absolutely fantastic. I don't know, anyone here? Yeah. And the people are passionate about it, like really, yeah, and we actually, we totally lucked out because we had one meeting with a government body, and we it was a little bit tense. We're talking to them about, what we're doing, and then I mentioned the cucumber thing, and he's like, I hate cucumbers. Like, I hate them. They're horrible. He's like, I totally get it now. And so it's one of the things that people can relate to. People can relate to the asparagus. People can relate to the cucumber. There's all kinds of things. And part of our goal is like, everyone can answer this question too. Do you hate cucumbers? Yes or no? So it's pretty easy to answer those questions. So this is, who can actually taste the bitter flavors? I'm linked to a lot of people. This is a lot of family members and random people who have shared with me. So this is where the sharing actually gets to be pretty fun, where I can see, this group actually can taste certain bitter flavors, and this group is not able to. So, it's a question, can I have a dinner party with this group and really serve these people something else? So that's how we do those. Oh, sorry, let me go back in the bit. So this is also how we start to link up with the 23andMe component, because a lot of people were getting this information and saying, well, why don't you ask me right away whether or not I like it? So I haven't filled mine out yet, but broccoli does not taste bitter to me, so I hit no. I do like black coffee. Do most beers taste bitter to you? Somewhat. And so now I can see my results. So this is the results of the 23andMe community. So all the people who have responded. And again, one of the things we've been shocked by, people really want to fill out surveys. It's just, and I think, again, we had a meeting with some government officials who were a little skeptical about this today. And it was funny because he was very skeptical about this. And the previous day, we'd had meetings with a few sort of Silicon Valley Web 2.0 buffs who were like, oh, all these websites about quizzes and Quiznos and Quiznot. I forgot what they're all called. They're all called Quiz something. But are doing so well and are such hot investment areas. And people are addicted to filling out surveys online. And then the government side was like, no one wants to do this. Like, no one's ever going to give you data. So, but what we found, people really want to fill out data. So here I can see how I compare to the community. And again, just simple kind of basic feedback that you get. So any specific ones that people want to look at? Any topics? I don't know if we have. Anyone have a favorite? Everyone have a favorite phenotype?
Earwax type.
Oh. The earwax isn't as sexy as it sounds. So earwax type is actually pretty clearly defined by whether you're Asian or not Asian. And I am in the wet, and this is our dry community. So although it's interesting, Linda's of European descent, and her father-in-law, who's also of European descent, has dry earwax. So he has a little bit of Alzheimer's. He's sick, but she's always kind of looking in his ears, like, ooh. So I mean, it's kind of interesting. Like, who knew? So here's a bit about earwax. Here are all the people I'm connected to. You can see I have a community of wet. I have a very small dry. Only Becca, who is she works at 23andMe. She's my only dry earwax friend. I have to tell her that. So I can show you also when the PRs look like. So we can look at things like nicotine dependence, which again this is where we get into a little bit of, we presented data. I don't remember where this study came from. So this is a study that was done in Nature. It is actually a big population size. It has 3 people, which means it was done in over 1,000 people. So right there, it's over 1,000. It hasn't been replicated, so I guess that's why there's no contrary studies. So we tried to give all the information so that individuals can almost look at this like movie ratings. And you can see here that I, on average, am more likely to smoke, or I'm more likely to have 1 cigarette today than the typical smoker. So, it's kind of interesting. I used to smoke when I lived in New York, and I have to say I really, really liked smoking. So it's one of the things that was interesting to me because I had no problems ever quitting, but it was, some people really seem to enjoy it, and some people really don't. And I don't know, these are the types of things that we want to be able to research. And if we could start to understand addiction and understand why some people smoke, why some people can't stop, it'd be useful. So then I can see here's the rest of the family in this group, which on average has less. So here are the citations, et cetera. So, yeah.
If somebody is having this kind of information about himself,
Mm-hmm.
then you could potentially use, if you're quizzing people, you could potentially use that information to try and study things. But if I see that you're showing me that I have a marker that shows that I'm likely to be more addicted to nicotine, my answer is going to change, right?
Right. So, and that is actually a fundamental problem. It's a question of can we overcome that with numbers, or can we overcome that by asking questions in the right kind of way? Is there a way, is there a way to do it? Because we've clearly biased you in some capacity. And one of the things that we say, we recognize that there's limitations to the kind of research that we do. We have debated in the company whether or not we should have you answer the question first and then show you the data. We're a little bit mixed, because from a consumer point of view, if you just paid money and I'm forcing you to answer a question, is that obnoxious? So, for some people, it might work. It's one of the things that we've debated a lot in-house, whether or not we should have you answer the question first. You could always skip the question. But I agree. It's one of the things that we, these questions on the side, the 23andMe component, we see that more as it's not necessarily going to lead to a publishable paper, but it will help us get preliminary ideas where we can then have a larger survey. So we've been working with the academic community to get validated surveys so that then we could say, okay, we're going to do a smoking study, and it would probably be a much longer survey. Can you actually have a variety of questions there where it's almost fraud detection, that you can try to pick out some of these biases? But it's a very good point. If you have a solution, let me know. Yeah?
So are you going through and hand annotating the quality of the studies and so forth? So that seems like that might have a problem with scale at some point.
It does. We have a big curation, well, not a big curation team, but we have a big science team, and it's one of the things, we can't curate every paper. So we try to keep track of, I think that we're able to do a very good job, but we recognize that we can't do everything. So part of the reason why we set up a scientific advisory board is that people do actually send us journals or articles if we've missed something, and when we do curate the main journals that are out there, and we do have a reading list, so everyone has to read the various journals that are coming out. But it's an amazing amount of work.
Right. So
but,
we have sort of a similar issue in our website of proteins, and one thing that we've thought about is possibly having some sort of a trust network where people can mark up what they think are the ratings of the paper, and it's something like Slashdot or they can mod up a paper, and based on how trustworthy we think those users are, we can have star ratings.
Yeah, we definitely would like to figure out a way that we can get the scientific community involved, so that we're not the ones who are putting the judgment as to is this a 2-star, a 1-star, or in established research. So we would love to find a way that the community actually can collectively curate. We've talked to a variety of major institutions that are looking into ways to potentially do that. No one has really taken the lead yet. So it's one of the things we've talked about as we are doing this. this might be something that we just put out to the community and say, here's the database of all the information that we've collected. We keep track of every journal and we index all the information that's out there. So potentially at some point we just make that available so that the academic community could participate and add more. The main thing that we worry about is quality, is that we just want to make sure that information is really input correctly, and that, and we would have to sort of work out exactly how would we do that. You could just have a journal rating system or paper rating system. But it's a lot of work for us. Yeah?
Can you zoom in even more and see whether the SNP is, where the SNP is, whether it's just a closely linked marker in the protein?
Yeah, so let's go back to another one, because it's always fun to pick a new phenotype. Let's see. So what you can do, let's take caffeine metabolism is actually a pretty fun one. So we can take caffeine metabolism. I can see, here's what the marker is. Here's the SNP. So you can see I'm a fast caffeine metabolizer, which is great because I really love coffee. Here's the SNP. And so it's actually then takes me here. So it lets me know exactly what, and I don't know if this is exactly what you're talking about, but it lets me know exactly where is it on chromosome 15, and it lets me know exactly what my calls are. So at some point, we'd like for you to be able to click on chromosome 15 and zoom in more and be able to see more things. Like, as more data is coming out, or if there's more interesting kinds of data, yeah, it'd be great to be able to start putting all of that in so you can really start to almost delve in a Google Earth kind of manner and explore your genome. So,
Any of the genome browsers?
say again?
Do you link out to any of the genome browsers like Santa Cruz?
We don't. We actually have almost, we've almost sort of replicated it. So this is actually, and I can show that now, this actually is our Genome Browser here. So instead of Santa Cruz, you can just use this. So if you have a favorite gene or SNP?
Well, the one we're just looking at.
So difficult. Let's pick another one. Anyone else have a favorite SNP or gene? We can always do,
CYP2D6.
what is it?
CYP2D6.
CYP. CYP2D6. So here you can see what all of the SNPs are that we're looking at, and all the different calls. So you can expand on these, and then you can get all the different reference looks. So it's, again, for most people, it's for any of you who plays around with this and you get your own data, it's a gold mine because it's really easy to navigate. For the average person, they don't know what any of these things are. So most people don't know what dbSNP is. It's a great interview question for us, if it screens out a lot of people. But you could click on any of those and then start to see. But this is one of the problems even with Google, is it doesn't have a lot of information on the SNPs. So, but you could click here, it's limited. This is something that we'll expand a lot more on. So the next area, I'm going to skip out of Gene Journal unless there's any immediate questions there. Yeah.
So you are just considering like SNPs variants, or do you have any structural variants?
We're only looking at SNPs right now. So I should actually clarify. We are genotyping people on the Illumina 550K, and then we custom designed an additional 30,000 SNPs specifically because we thought they were interesting. So we looked at the whole HLA area. There's something called DMET, which is distribution, metabolism, excretion, toxicity. So because we're so interested in pharmacogenomics, what are the genes that might be associated with drug metabolism? So we specifically chose SNPs that we thought were going to be interesting or meaningful. So it's 580,000 plus the 550,000 plus the additional 30,000. So there's limits. We'll be able to do copy number variation at some point, and clearly this is going to move towards full sequencing. But right now, genotyping technologies are getting to be so cheap that it's more a question of how can we get, when this becomes an iPod pricing, you can get huge adoption, and then you'll get a huge data set. Mm-hmm.
How much does it cost to run one individual sample, and how much do you charge to run it?
So we charge $1,000. We can't disclose, the full margin, but I think anyone have a lab who knows roughly how much a 550K costs? I I mean, I think it's you know, the 550K plus the custom pricing plus processing, it's not a huge margin business right now. And that's not actually what we see as our long-term business goal. Our long-term business goal is that there's opportunities once there's a very large database. So there's ways then to actually start to connect people with the research world, that we think that that actually is a bigger value than just charging people for their genetic information. So again, it's a business. Like, we have to make money and we have to sustain ourselves off this, but it's not an egregious, you know, it's still relatively expensive.
In the long run, you consider selling access to 1 million people worth of information?
So we would never sell access to your genome, but what we might do is say, let's say you have connected and you said, you know, I'm really interested in caffeine metabolism, and I'm really interested in, you know, I have knee pain. And and let's say Pfizer's doing a study. What we would do is potentially facilitate that connection and say, you know, you said that you were interested in a study about knee pain. Do you want to be contacted? And so it's more, you know, we're really sensitive to the fact that this is your information. You have the right to control it. You might say, I never want to be contacted by anyone. So, but part of our goal is that if you're an individual that really wants to participate in research, then we want to enable you to do that. So, you know, again, part of our goal is we always have to balance that, because the end goal is to facilitate more and more research. So we have to always weigh, you know, we're not going to charge egregious fees on the other side either, because we want people to be accessible. Yeah.
So that said, from the standpoint of research and having a broad data set and a non-biased data set, how concerned are you that, especially at the beginning, your database is going to be very biased, both in terms of economic and educational status? Because certainly a huge portion of the population is so fearful of science that they would never agree.
Yeah. Oh, sure. I mean, it's just one of those things. Like, it's just a bias. It's like it's a bias that we can, I don't know if we can quantify it yet, but we recognize that we definitely have an intellectual, wealthy, you know, consumer base right now, and we can see it's not reflective of the population in terms of diversity at all. So it's, you know, we need to take on programs, and we need to do things. We need to drive down the price, you know, also to make it more diverse. But it's one of those things, it's just a limitation of what we can do. So our philosophy is that we can still do research on it. It's just we need to know what those biases are. Yeah.
So can you download in bulk the entire your entire data and that of your friends that have allowed you to see their data? So
no, we do not let you. So you'll see here, I'm only connected to Josiah, who is one of my relatives whose account I control. So I cannot. So I can download my own account, but I cannot, like, even my husband, I do an extended share version with him, and I cannot download his data. So you have two options of sharing. You can either share in an extended mode where I can see all the gene journals, or you can share just where you're looking at the ancestry and kind of some of the fun traits. But so you have those two options, and you can make that decision when you decide to share, and you can cancel the sharing at any time. So no, we intentionally prevent that. Yeah.
So on a sort of similar vein, it seems like one risk of having your service is that if information about your DNA ever gets out, and you know you're all of a sudden susceptible to heart disease, then you might not want that to be public information for insurance purposes, things like that. So kind of what assurance? I don't even know how one would begin to quantify that, but.
Yeah, so right now you can sign up for your account and I should show you one of the people I share with. You can sign up, you know, you can have someone in your lab buy the kit for you and sign up under a different name, and I would never know that was you. And I also, we'll show you in the caffeine metabolism, you know, you control your data. So if you don't want to share with anyone, you don't have to. And 23andMe is never going to make your data public. Like we're just a service to allow you to do with your genetic information what you want. So it's accessible online, but we won't make it publicly available unless you tell us to make it publicly available.
So are there specific legal restrictions that, you know, the user retains the right to your data and only sort of licenses? Basically, are you legally allowed to share the data? Or
you mean legally in terms of California state law, or in terms of our consent form?
If you give the data to someone else, can someday, could I sue you for?
Yeah. I love those questions. It's great.
And if so, for how much?
Yeah. Ouch. No, I was kidding.
What I mean is, you know, this is really fascinating for me.
Yeah.
But I'm reluctant to do it because without sort of a particular legal covenant.
Yeah. I would encourage you to read the consent form. The consent form is when we went to the World Economic Forum, we actually had Larry Summers read it with, I don't know, it was like an hour and a half. It was torture, and so it was, you know, going through every detail. But the consent form in and of itself took us almost 9 months to develop. So with so much back and forth, because the company will not succeed if we can't protect your privacy. And we worked with a number of privacy groups to try to make this as secure as possible. The limitation that we made, one of them suggested actually setting it up so that you could just send in a money order. And you could set up a P.O. box and you could have a money order, and then we would never know. But that market is so small and those people are so unlikely to do our tests that we decided not to do that yet. But for the most part, like, you can sign up, for instance, this is what I was showing, you know, DG Party 7. You know, I know who it is, but because it's a friend, but it's not, you know, you can sign up under any name you want.
Yeah, but even if your name is hidden, if you're sharing your information, somebody could potentially figure out that you're closely related to somebody else.
But you don't have to share.
Okay.
Yeah. It's all an option. And it is one of the things that comes up where people have, you know, one of our lead product developers shared his information with someone else in the company, and then that other guy in the company came in the next day and he's like, oh, you know, you have DNA in common with so-and-so. And he was like, oh, really? That's weird. Like, so it is, I mean, it's one of the things we disclose that. If you're sharing, you might learn things that you didn't know, but then don't share, or just share very selectively. And so, but again, it's part of goes to the fundamental premise that if you want to share, you can, but you don't have to, at all.
But isn't one of the problems if my brother share and I don't want to share?
But that goes to sort of a fundamental question about your genetic information in general. I mean, one of the issues that comes up is paternity. You know, you and your brother could get genotyped and find out that you're half siblings. So, and then that. And that then, you know, and that's, we've had one person at least write in about, you know, saying that that's happened, but they are still happy with the service, but but there was a surprise. And that actually, I mean, and that's one of the things, like, it's a fundamental part of your genetic information. Like, there's not a way around it. So there's, you know, you have the option to say, you know, we don't accept samples from families, but, you know, part of the fun of this, and I should get to it, part of the fun of this is the families. Let me actually go to family inheritance, where, you know, so it's one of those things, it's definitely a question, and it's not for everyone, and we don't try to push it on everyone, but it also becomes, one of the things that we found is that if you had a child who was autistic, you just stop caring about a lot of HIPAA privacy issues. Like, your goal is, am I going to have another autistic child, and what could I do to help this child? So it's a balance. You know, there's always a risk with everything, but the potential for good, we think, overwhelms that potential risk. So this is the Genome View. This is a way that you can actually start to compare, and you can actually see what DNA do you have in common with other people. So this is me compared to relative, but it's not showing up. Why is that not? So I'll compare you to my sister. We have a lot of random people in here. And so you can see here, so where it's dark blue is where we are identical, and where it's light blue, we're not phasing yet. So where it's light blue, I know I'm half similar, and where it's dark blue, I know I'm identical to her. So I know we're sisters. And the thing that's fun, like, people look at this and they say, well, like, oh, so what can you do with it? And this is an area that we'll really start to expand on, because as you start to know more and more about all your chromosomes, you'll be able to zoom in, zoom out, learn things. I was able to learn, I have another sister who had found out that she was pregnant. She found out she was a carrier of Bloom's disorder. And so I, of course, go online, I look at Bloom's disorder, and I find out it's gene BLM. I go in and I type BLM, and I see BLM. Come on. BLM shows up right there. So on that little triangle, and this is my other sister, but I was in the dark blue with her, so I knew that I was also a carrier of Bloom's. So I go into my doctor and I tell him, like, oh, I'm a carrier of Bloom's, and he's like, how do you know that? I'm like, oh, because I did a genetic comparison of the two of us, and I saw that I'm identical to my sister in that region. And then he was like, well, why don't we do a validated test? I mean, this is where it gets to be, it gets into being issues. So you can imagine for areas like BRCA. So I also did the BRCA test, and I, just because I do every test that's out there. And so I essentially then tested the whole family. And, one, that's a question for Myriad, but two, it's also a question for my siblings. Like, did they want to know? So these are all the exact issues that we are grappling with. Like, this is a question of how society is going to change, because what I find out about myself is going to impact my siblings and my parents. So Linda and I are both thankfully from families that are okay with being very open. They didn't really have a choice. So the thing that's also interesting is you can then look at where is the immune system compatibility. So this gets into your HLA region. And this is a question Linda and I just personally have, because Linda has non-identical twins who are, identical right on chromosome 6, right in that area. So it's a question, would they potentially be good organ donors? Is this a new way that you could potentially match people other than just doing HLA typing? Could 23andMe actually be something that actually helps connect people? And it hasn't really, the studies just haven't been done, but I think that there's that potential where, could we work with the Red Cross in some way? So the thing that's fun is that we've been able to find, I'll just have to show you because it's kind of cool. Let's see, it's my mother-in-law and my, what's his name? My grandfather's brother. See? They have a little DNA in common. So I'm Jewish on my mom's side. He's Jewish. We're both from that same area. Kind of supports that theory of small communities, slightly inbred. But we are kind of excited, like, oh, that's kind of cool. So we have this, again, we don't really know what that means, but people really like it. And we changed the thresholds on this system to, to actually have it be only the bigger chunks. And all these people rebelled, like, no, I really wanted to be related to more people. So it's one of the things that we work with. We also have this view, Grand Tree, which I'll just talk about. I won't go through it all, but you have the ability then to put in your grandparents, your grandparents at the top, the parents, and the child, and you can type in any gene, and you can actually see specifically who did you get those genes from. So it's actually pretty cool to be able to see, who did you get this from? You can put in multiples. So a lot of Linda, I don't have all 4 of my grandparents, but Linda, my co-founder, has all of them. And for her, it's, anytime something interesting comes out, she's like, oh, I want to know who I got it from exactly. So I can switch over into the Ancestry side, which is where you get your maternal. I see I'm running low on time. So I have my maternal ancestry. So, and this is something people are just people are obsessed and fascinated with this area. It's just, where are they actually from? You can see here all the people I'm connected to. I can click on any of them and see. Or maybe not. Let's get an interesting one. Oh yeah, where is Mike? Is he in there? And oh yeah, N1B2. Let's see what Mike is from. You can all tell him, you're an N1B2. So here's Mike. So here he goes through here. It's his history. And then we have a haplogroup tree, but the haplogroup tree has been a little bit difficult to navigate for people. They just haven't fully, it's a tough concept to grasp, this mother of all mothers. But you know, the history, the map, and people just, I mean, we've been amazed at how much people love it. My husband, I have to show, let's see, I'll show you on the paternal side. So then we get to the paternal lineage. And this is just looking at your Y chromosome here. I don't have one, obviously, because, which is refreshing. But you know, I can look at everybody else here and see, here's my husband. So I can, if I have a boy, I'll know, this is my husband's, he's a K2. And that's roughly, I mean, it's pretty interesting for me that that's roughly where he's from. So that's where 500 years ago, that was the prevalence. Yeah?
So if you're a potential power user of the system, would you be interested in actual numbers?
Say again?
Would you be interested in actual numbers?
Actual numbers of what?
So when looking at Ancestry, I'd be interested in concurrent systems.
Oh, yeah. Yeah. Oh my God, people in Ancestry area are really sophisticated. It's difficult. We don't get nearly the same kind of sophisticated questions on the gene journal side, but Ancestry, there's so many sort of novice professional genealogists who ask really sophisticated questions. So one of our next release cycles is to put out a lot more information. So we're not giving all the numbers right now. But we will actually allow you to dive into it a lot more. Ancestry is going to get a total overhaul because we've recognized that the community is far more sophisticated than we realized.
I can see a website, a service like this kind of bridging from a lot of different sources.
Mm-hmm.
Do you see an advantage of this having come from the for-profit sector? Like, is there something that like a direction it's taken or a direction it may take that, as opposed to having come from either a nonprofit organization or an academic group or some other system?
I mean, I think the advantage is that we know how for-profit world works, and that, you know, part of our goal is to really merge nonprofit, for-profit, and consumers. And knowing how for-profit works, I think, has that advantage because it's its own unique beast. And you know, hopefully, coming from it, both Linda and I, that we'll be able to integrate better with it. So let me just show you the chromosome painting very quickly. Ancestry painting. And this is really where you can start to break down your genome and see what percentage European, Asian, African you are. I'm very boring, but because I look almost all European. I'm 99%. But this is my roommate from college, who is our model child. I was like, what a great roommate. She is, her father is African American. Her mother is part Native American and part Mexican. And her grandmother is from China. Her great-grandmother is from China. So, excuse me, she clearly has such a diverse background. But to me, what's really interesting here is that she's always associated more with the African American community. And when she saw this, she originally was like, this is like, why would I ever want my genetic information? And now she's obsessed with it. Like, oh, this is so cool. Like, it kind of changed the identity. And one of the areas we really want to do research on is exactly that question. How does this change your identity? And I was at a coffee shop on the weekend trying not to eavesdrop on these two guys, but they came down and they sat down next to me, and it was unbelievable. They sat down and he's like, you know what? I just got my genotype results, and he was talking about, and he's like, he has a very Jewish background. I feel so close to him now. A very Jewish history, and he was clearly dating someone who was non-Jewish, and it was that question as to, well, what is his Jewish identity? What does it actually mean? What would it mean for his children? And then they got in this whole dialogue about what is the definition of race. And I was saying that, like, this is unbelievable, like how these people are just like talking about this. Like, it was unbelievable that this was actually, like, all the things we thought it would impact, that it was actually impacting these people. So it really, I mean, this is an area we're eager to do a lot of research on, is how does this actually start, how do people start to view themselves? You know, how is race actually going to be defined? So let me go quickly into the 23andMe area. I showed you a tiny bit of it, but this is where we're hoping to do a lot more of the actual research. This is where we work with academics to put out validated surveys. And this is one that's out right now on handedness. And you can see it's not just a question of are you left-handed or right-handed. I already filled out my survey, but I am right-handed, but with a weak left preference. And 17% of the population is like me. But you can see only 55% is pure right-handed, and a very small percent is really pure left-handed. And this is about 15 questions. I forgot who. Yeah, here's the survey citation. So we've been working with a variety of different academics in terms of, if there's a survey that's out there that's been validated, it's fun to put up online. People want to fill out this information. We're happy to give information back to, aggregate information back to the academics. And then people love this. Like, I never knew that I had a weak left-handed preference. We can go back then here. So we have a featured survey usually, and then there's this 10 things about you. I'll show you what it's like. And again, we've started more now with some of the sort of fun phenotypes. So do I have a tendency to sneeze in bright sunlight? No, but do a lot of you? I had no idea about this. I wanted to cancel that question, and there was like uproar. I am naturally a night owl. When I clasp my hands, my left thumb, you guys? So again, we don't know if this is genetic or not. I mean, it's hypothesized. I go to the next page. And then I do, unfortunately, wear contacts. I am a dental superstar. I think that I'm right-handed. I have some, not a lot. I like both salty and savory. This was a controversial question. And the asparagus. Yes. It's so great when people don't know, and they're like, do you have to really dig down into the toilet? And I have slightly wavy hair. And then I can see the results. And again, we've been surprised. People are just addicted. People love doing this. A really high percentage of our customer base has been filling out these surveys. So it's one of the things that we wanted to see how people were going to respond, but we've recognized that we actually need to put more and more surveys out there. We're cautious. Some people wanted to put, some of the behavioral ones are a bit, we're a bit cautious about. But here's then the results. Do you have a tendency to sneeze? No, but a pretty large percentage of people, I mean, much larger than I never even heard of this issue. But 32% there, and then night person. Most people seem to be night owls. Clasping the hand is surprisingly pretty equal. I'm interested that there's a small percent that can do either.
How come you can't see the total number of respondents? Whether that's 10 respondents or 10,000?
Yeah. So we don't do that really, actually, for competitive reasons. Is that we don't talk about the size of our database, and at the same time, we will at some point start doing things like that, because when we have more specific studies, we might say, we're doing a study on Crohn's, and we need at least 1,000 people to participate in this. And so then we would actually almost have it like a fundraiser type of thing, saying, you're the 353rd person who's participated. We need another 700. So we might do things like that, but we have a limit. So we have set sort of a threshold where we won't show results until there's at least a certain number. To, again, worrying about the privacy issues is that we want to make sure that whatever we're doing, we're not potentially violating someone's privacy. So, but I agree. I mean, numbers are clearly interesting. Yeah, we've had. Yeah, I mean, there's some. I mean, I don't know how often we're updating it right now, but it's. You could potentially start to calculate things. But again, we worried about that. We don't want to put in. We don't want to allow you to figure out the exact numbers. So here's, again, I was surprised. 30% of the population. It's hypothesized in the teeny bit of academic literature out there about the asparagus that it was only about 10% of people didn't smell it. But we're seeing actually a lot more people. So again, NIH is not going to fund this study, but we might. Yeah?
So presumably the part of the questionnaire is look for
So we actually just came out with this probably 4 weeks ago. So we're just starting to actually do that. Clearly, and the nice thing about being in the for-profit area is that we want to maintain an academic reputation or a scientific reputation, but we're not so focused on publishing. So we're happy to just self-publish this stuff on our site. So as this stuff comes out, we want you to be engaged. So, and we'd love that. And if other people end up replicating it, that's great. But we'll always have the surveys out there. We'll always keep, updating the data. But when we do get results, we should probably just publish it on our site. Yeah.
I noticed at the beginning of your presentation, there's a section where you could look at a list of different links to articles. For the public that isn't in academia, is there any ways for them to access those articles through your site, or is it have to pay?
I mean, it's a huge problem. No. I mean, we try to make sure, that's why Plouse is great. But no. I mean, it's just, it's a really sad aspect. The fact that, this is all government funded and you can't access it.
What was the question?
Is whether or not, if, in the Gene Journal section, we link to all the papers, but most, if you're not in academia and you click on the paper, it's going to tell you to pay $25. So, that's definitely, most people won't understand it anyways, and they're definitely not going to pay $25. So it's unfortunate. We would love to be able to link to the real papers. If an academic has put it on their website, we will try to link to that site. So we actually do check. So this is the main part. One of the things, the direction of 23andMe really is focused on having more and more surveys that are out there. We're hoping to launch very specific projects. So if there's very specific phenotypes that are of interest, autism is something that's always come up as something that's very, it's of huge interest out there, but it's so complicated, so it's not going to be our first project that we take on. But this is where I think that 23andMe will really start to do a lot of academic collaborations. So they can just then go through, again, when we think about sort of our main challenges, I'll just leave in this format. When we think about sort of our interesting problems, we're dealing with data sets that are bigger than actually most studies that are out there. So we do, routinely we'll do sort of all versus all comparisons of the whole data set. And it's getting, not a lot of people have tremendous experience dealing with thousands and thousands of genotypes. So it's going to become more and more of a challenge over time when we start collecting a lot of this phenotype information, and then we have 580,000 data points about people. It just becomes it's going to become a huge statistical problem for us. One of the things that comes up a lot is that this really is a new way of doing association studies. And I think the point you brought up even about the biases, there just are biases in this data set. And how can we work with that? And how can we actually grow this to be large enough so that we can hopefully factor out some of those biases? Or if we can't, how can we just start to work about it? So there's limitations to what we're doing, but we think that by having lots of information, we can probably overcome some of that. So would you rather have a perfect study of 500 people, or would you rather have 100,000 people with some dirty data? So we are going more of that approach. Bigger data sets is really what you need. So again, but it's a big challenge for us, and we're not going to get it right the first time necessarily, but we need to keep evolving. Collecting phenotype information is actually a really fun problem, because it's one of the things that Web 2.0 community, the whole internet community, has really realized. People want to put information out there. What is a fun way that you can actually start to collect that information? And developing surveys, developing fun tools. People do really dumb things all the time just to get a little bit of information back. Or you look at Facebook apps and MySpace, and people really want to put information out there. So what are creative ways that 23andMe can come up with to solicit information out there? And Louis Van Ahn, I don't know if you know him, he developed the really brilliant program for Google. He developed it on his own, and then Google licensed it, where Google had this problem where they had all these images and nobody was annotating the images. So how are they going to, they can't hire enough people to really annotate all the images. So Louis Van Ahn had this game where you put up an image, and you get 2 random people who are on the internet, and you type in words that you think describe that image, and as soon as you have a match, it moves on to another image. And so it's a 60-second game, and you try to see how many images can you go through. And Google's assumption was that if you have 2 people who both put in the same word, then you just annotate that image. And so it was a really brilliant way that and people were addicted. People were, like, some people were online 7-8 hours a day doing this game, and that's the beauty of the for-profit world. No one knows. So what are ways that 23andMe can actually develop fun games like that where people want to input information about themselves, and that will be helpful to research. And then the last area is that we're clearly moving towards full sequence, and full sequencing and copy number variation and all kinds, again, we think about all kinds of other data that we can start to collect. It's just going to become monumentally more challenging for us. So we have a team of almost 20 scientists now, anticipating that that's actually really going to have to grow, because if we are being seen now by a lot of groups as, could we actually start to become a hub of data analysis, we're going to have to really start to grow this. And as our data set grows, over time we're going to get, higher density, and we're going to get cheaper, so volume's going to keep going up. It's going to become more and more, it's going to be more and more challenging. So on that, I'll see if you guys have any more questions.
So you mentioned that you want to bring in researchers in this. So how are we anticipating sharing the data?
So sharing the data is really a challenge. What we would do is we could do partnerships where either you could say you have a specific set of queries that you're looking for, or you have a specific hypothesis in your mind and that you want us to actually run those queries for you, and we send you back the aggregate information. Or we can't let the data leave our site. We have the potential to say, you could come on board, you could come on, and we say, we do some kind of 2-week fellowship or 2-week program where you just come on and you work with us on it. So again, our main concern is always protecting individuals' privacy, but a lot of those individuals who have inputted phenotype information want academics to get access to it. So right now, we'd probably, either you send in that you have specific queries that you want to put in, or you actually come in and we work with you on a specific project.
So follow-up question. In the academic world now, everything should be publish the data, publish the data.
Yeah, and so what we could do is when we take on new projects with individuals, one of the things that we could do is we have another consent form. So with the Parkinson's Institute, we actually have another consent form where genotypes are going to be housed at Parkinson's Institute and at 23andMe. And so if we took on a large enough project actually with a specific group, we would say, data would be housed here and data would be housed at 23andMe. Yeah.
I have a question. You keep commenting that you're a for-profit institution. Is all your profit from the $1,000 you charge for that?
Right now. Right now it is. I think that there's clearly that potential. Again, and part of the reason why I showed the numbers early on is that $60 billion a year that goes into research is that a lot of that is inefficiency. And I think that we could cut down that number, drive efficiency, and I think that we could be a major part of that. So
we actually want that number to go up.
Yeah. Yes. Well, I would rather have that number go up, but in a better way. But right now, if you talk to a pharma company and they say, "Oh, I want to do a study of," we talked to a group that is doing a type 2 diabetes study, which should not be hard to enroll, and they're only looking for a few hundred individuals, and they said, it's going to take us 18 months to enroll the study. And for me, it's just like, how could you not enroll that quickly? And that's why I love actually. I spent a lot of time in India, and they could, they would say like, "Oh, well, we could enroll the study probably in 2 days." Like it's so different. So I think that one of the things that we could really do is help with, trial enrollment and really start connecting individuals. So there's other business opportunities there, but right now it's just based on this. Yep, you had your hand.
What is your policy for children under the age of 18?
So you can't consent yourself, but you can. If you're a parent or a guardian of someone, you could submit their DNA for them. This is where it gets into a lot of the tricky laws, and I'm not the legal expert, but I think you can share it, Again, if you're the mom of a child and you want to share it with your parents, you would have that ability to do it. If the child was under 13, I think you can't post any of that information.
But you can still share it.
You could still share it. So it does get into some of the, it's a controversial area. We get into people saying, what should be those rights. But again, it's like a guardian. So if you had a 10-year-old child and you wanted to test them for something else, you would have the ability to, and you could share it with anyone you wanted. So we see it's kind of in that same range.
So let me suggest that since Brian McClendon has created a nice reception for us outside, that we stop the formal part now and continue the discussion of reception. And thank Anne for a nice